Sorry. I guess we can. So for this week, we're just going to be reviewing topic 9, which is DNA replication. Okay. So first, just a quick recap on DNA structure. There's 4 bases. They base pair through hydrogen bonds. There's phosphodiester bonds that link the nucleotides together. And then for the actual structure, it's just a base pairing of 2 strands together, which creates the double helix. And even as a single strand, DNA will form a helix. And the strands are in opposite complementary direction. So one's going 5' to 3', the other's going 3 to 5', which is also known as anti-parallel. And then the 5' end is just where the 5' carbon is attached to the phosphate, and the 3' end is where the 3' carbon is attached to the OH group.
So this is just a little bit about the nucleotides. So they're the building blocks of DNA. They're made of this 5-carbon sugar. They have a phosphate group, and then the nitrogenous base, which is variable between the different nucleotides. And then those nitrogenous bases are able to base pair with each other through hydrogen.
Okay. And then a little bit more about the structure. So it's just 2 anti-parallel complementary strands. Each strand is made up of nucleotides. Each has a 5 and 3' end. And then the bases are paired together through hydrogen bonds. There's 2 between adenine and thymine, but 3 between guanine and cytosine. And then it forms a helix with a major and minor groove. The minor is the narrow, non-specific backbone interactions, whereas the major has the sequence-specific backbone interactions.
And then just like a magnified version of nucleotides with that phosphate group, the sugar, and in DNA, there is an OH off the 3'. And then the different nitrogenous bases in the 4 different nucleotides.
And then this is just another enlarged detailed view at deoxyribose. So the 1' carbon is attached to the base. The 2 is attached to a hydrogen, which in RNA would be attached to another OH group. The 3' is attached to the hydroxyl group, 4 hydrogen, and 5 is the phosphate. And then this is just like a little acronym to remember which are the purines. So purines, gold, adenine, guanosine. And then the other 2 are the pyrimides.
So a little bit more about base pairing. The bonds between the nitrogenous bases are hydrogen bonds. Adenine and tyrosine have 2 hydrogen bonds, and guanine and cytosine have 3 hydrogen bonds. So the more hydrogen bonds, the harder it is to break. Just remember that. And this is talking about why it doesn't, like the double helix doesn't use covalent bonds to hold itself together. Basically, those strands need to unwind. Helicase needs to be able to come in there and separate those strands. If those were covalent bonds, you wouldn't be able to do that with just DNA polymerase. So they need to be hydrogen bonds in order for DNA to be
Okay.
replicated.
So DNA is synthesized from 5' to 3', which means nucleotides are always added to 3' end. And when we add nucleotides, they must have 3 phosphates. And then these nucleotides are held together by phosphodiester bonds, which are covalent bonds. So in the red box, oxygen from the 3' hydroxyl attacks the phosphate in the first phosphate group, which is a nucleophilic attack. I don't know if anyone's taking orgo, but talk about that a lot. And then the second step, it causes the bond between the phosphate and the oxygen, which is in blue, to break up the other two phosphates. And then that's what forms the phosphodiester bond between the 3' carbon and of the sugar of one nucleotide and the 5' end of a sugar on the other.
So DNA follows a semi-conservative model, which means that half of DNA is always conserved. Yeah. Yeah, like they dissociate. So, for DNA replication, parent strand is separated by helicase, and then each of those strands serve as a template for DNA replication. So half of the DNA is always conserved through replication. That's basically the semi-conservative model that DNA is.
Okay. And then next we'll move on to DNA polymerase proofreading. The proofreading function increases the error rate to 1 mistake in every 10 million nucleotides. This is provided by an addition of a domain, a subunit, both of which that have exonuclease activity. The DNA polymerase is able to sense where a mistake is, move the incorrect nucleotide to the exonuclease active site, which has a 3' to 5' directionality. And it'll chew back to the 5' end. The strand will move back towards the exonuclease active site to add the correct nucleotides. And the process of DNA polymerase proofreading, if we go back, goes along with this idea, because if it was the other way around, it wouldn't like cleave the phosphates, if that makes sense. It needs to break it apart in order to be added, but if it was reversed in this situation, it would already be adding to where it only has one phosphate. I don't know if that confused anyone, but I remember her talking about that in lecture when I took the class. Do you want to?
Yeah, just a little bit more about proofreading, a little bit about like how it happens. When the DNA polymerase senses that there was a mistake made or a wrong nucleotide was added, it'll take that nucleotide out of the active site and switch it over into the exit site, so that it can cut and cleave that nuclease off and fix the mistake.
Yeah. I think it's just that a mistake has occurred. Like if it's adenine and it adds like another adenine, it knows it's wrong, type of thing. I don't think it. I think, I can't remember off the top of my head. I could try and figure it out after the slide, but I think it knows which was the newly synthesized one. But just remind me if I don't remember to get back to you after the presentation's over. Okay, so next to replication, at the origin replication, that's where the cells start replicating their DNA. In S phase, cells have multiple origins of replication, as our DNA has billions of base pairs, and then not all the origins will fire at the same time. And it's important to keep in mind that replication occurs bidirectionally.
So this is just a little bit about the replication fork at the origin of replication. There's 2 replication forks at each origin of replication. One is going to go to each side because replication happens bidirectionally. So in the next slides, we'll go like a little bit more in depth about what each of these branches are. But yeah, you should know that there are 2 replication forks. One goes in each direction, and yeah.
Okay.
So this is just a little bit more about each of the proteins at the replication fork. Oh, and then not in this picture is a sliding clamp loader, which is an ATPase and uses energy to open and close to load the sliding clamp onto the DNA.
Also, it's important to note that the sliding clamp itself is not a ATPase, just the loader. And then this is just another list of all the definitions of what they do, similar to this. Okay, so then you have your leading and lagging strands. Directionality is really important, so make sure you pay attention to the strand you're making and the direction of that fork, which will determine whether you are leading or lagging. I would recommend always drawing it out yourself. When trying to solve problems, it'll just be a little bit easier. And then if you're synthesizing in the 5 to 3' direction of the fork, you're leading. And then if you are synthesizing 5 to 3' in the opposite direction of the fork, you're the lagging strand.
So this is just some information about the lagging strand. The lagging strand is always going to have basically Okazaki fragments, which we'll talk about more in a few seconds. The lagging strand will always have Okazaki fragments because it is not continuously being synthesized. So if the DNA polymerase binds, it won't just continuously synthesize the entire strand. And that's because the fork is moving in one direction and the strand, like the parent strand, is not in the direction of synthesis. So just always remember that your lagging strand has those Okazaki fragments and it's not continuously synthesized throughout DNA synthesis.
Okay, and then when determining which is the leading or which is the lagging strand, you want to always first start that new DNA strands are always synthesized in the 5 to 3' direction, and the direction of the forks is starting from the origin and moving outwards. So what you want to do is compare the direction of the fork with the orientation of the parent strands to determine which one will be the leading and which one's the lagging.
And I think this is just an example that we're giving. So here we have 2 strands and we want to talk about how to determine the leading versus the lagging. So we're going to look at the replication bubble and then you're going to think about what's happening to the right and then separately think about what's happening to the left.
So to the left side of the origin, so DNA can't be synthesized continuously. If DNA was, then the resulting DNA would be parallel to the parent strand. We could see that the top strand must be the lagging because it needs to be synthesized against the direction of the fork, and then which creates the Okazaki fragments. Do you want me to continue? Okay. And then looking at the bottom strands, we see here that DNA can be synthesized continuously as it is going in the 5 to 3' direction. And so it's the leading strand.
And then you think about what's happening to the right of the origin and then compare it to the top strand. And you see that since the top strand is synthesized continuously, the DNA polymerase binds once and is able to synthesize the entirety of the parent strand. So that must be your leading strand. And that's just because of the orientation. And then you look at the bottom strand. And since there are these Okazaki fragments and the DNA polymerase needs to bind and unbind in order to synthesize the DNA, it's going against the direction of the fork, and therefore it is the lagging strand.
Okay, and then next we're gonna go over the N replication problem. Okay, so what exactly is the problem? It's caused by the lagging strand. When the primer gets removed from the last 5' gap, there's no upstream 3' end for the polymerase to bind to and fill in the gap. And this leads to the shortening of the genes over time.
So telomeres are structures at the ends of chromosomes that are related nucleotide sequences. And this way, chromosome can lose these nucleotides without actually losing the structural gene. And then telomeric proteins bind to the sequence and link the DNA to protect it. How like we're able to replicate our DNA without losing structural genes.
And then telomerase is an enzyme, sorry, that carries its own RNA sequence. It extends and lengthens the template strand. RNA is used as this template for the DNA polymerase to come in and continue adding nucleotides to the 3' end of the lagging strand. And telomerase is has reverse transcriptase activity. And this extension is what allows for the primer. It acts as a primer, sorry, for DNA polymerase to finish its job. Yeah.
Dissociate. That RNA should dissociate just like a primer does, like in the actual DNA.
Yeah. Well, I imagine it has to get removed because the normal primers get removed. You can't have like RNA, DNA, because that's very unstable. So I think it does get lengthened, but I don't think it codes for anything necessarily. And then if there's no questions, we just have, I think it's 3 practice problems. So this figure is here showing a strand of DNA being replicated over 3 time points in S phase. And how many origins of replication are there? How many origins of replication are there in the strand of DNA shown? Does anyone have any ideas? Yeah, it's 4. I'm using my calculator. 1, 2, 3, 4. Okay, so first, looking at this for number 1, would you guys think it's the 3 or 3' or 5' end? Does anyone have any guesses? Well, we could kind of also answer number 7 at the same time. So you can see it's not Okazaki fragments, it was synthesized in like one go. So then that means it was synthesized in the 5 to 3' direction, meaning that the end has to be the 3'. And then, yeah, that's deleting. What's the next one? Okay. Number 6, what enzyme joins the nick or gap in between the Okazaki fragments? Yep. Yep. And what else? Number so, what is number 4? Yeah.
Anybody have a guess? what these are. Yeah. They should be the primers for DNA polymerase.
Oh, and then number 5, I already answered. Those are just the Okazaki fragments. Okay. And then just some true or false. So for A, each time the genome is replicated, half of the newly synthesized DNA is stitched together from Okazaki fragments. True or false? True. Okay.
True. Just because every time the parents can be separated, one of those strands are gonna be synthesized through Okazaki fragments, so that ends up being.
B, when bidirectional replication forks from adjacent origins meet, a leading strand always runs into a lagging strand. True or false? Yeah. Okay, and then lastly, if an origin of replication is deleted from a eukaryotic chromosome, the DNA on either side will ultimately be lost as well because it cannot be replicated. True or false? False, yep. And then there's also a problem about that, I'm pretty sure, on the problem set. Okay. Just another
Yeah, I think this
overview.
is just to show like why the last one is false. Because at every like origin of replication, you have 2 forks going in each direction. So at some point they should meet up in replication. So we should include that. Say that again.
Needing another piece of
And then that's it for the PowerPoint. Does anyone have any questions? Yeah. The proofreading, right? Okay. Let me stop recording.
What?
I think I did. Did I?
Oh,
I think. Let me see. Oh no, it is still recording. Just kidding.